So welcome to topic 6.7. We are going to be covering mutations in this topic. Something we've talked about before in evolution is the source of all variation. Without it, life couldn't exist as we have it today. We need this variation. And today we're going to talk about it at a more granular level, kind of at the level of these of the individual bases and things like that. So, what are some sources of mutation? Well, there can be errors in this replication process up here. Right? If we have an error in replication that doesn't get repaired correctly, which happens quite often, that can cause a mutation. Chemical mutagens can cause a mutation. They can cause different bases to be inserted, deleted, swapped out. Radiation can do the same, right? Radiation, typically, the one that we most often associate with this is UV radiation, right? Sunlight is a source of radiation, which can cause mutations. And of course, mutations can alter this process, right? If we have something mutate up here, that's going to affect things down here, which could possibly affect things right here. So this is a disruption of this process of DNA to RNA to protein, or the central dogma. It can be a disruption. So let's take a look at some DNA and see some different ways that things can be disrupted. You can have insertion, which is the insertion of a base, deletion, deleting a base, or you can have what's called as a point mutation, which is a swapping, add, subtract. I'll use math terms here. Swap. And this is going to create different types of problems with its DNA, or possibly no problem, as we'll see. So, for instance, if I talk about an insertion adding a base, all I mean is that we're saying in here, oh, we have a mutation that caused us to add a base right there. I'll put an A between there. So, is that going to affect what happens here? Well, yes, it is. This is going to cause what's called a frame shift. Frame shift, which is a shift in the reading frame, which means before we had, if you look at the DNA in these three pieces here, these triplet pieces, you had TAC. Now you have CCA, whereas before you had CCG, and then your next triplet would be GGT. Your next triplet would be ATG, et cetera, et cetera. So this definitely causes a shift in the reading frame. So how's that going to affect everything? Well, this should be the same, right? But what about the second codon over here? GGA would lead to GGU over here, right? And then, so because we had a shift in our frame here, that's going to change other stuff too, right? So GTT is going to be our next three, right? CAA, right? Before, our triplet was AAU, right? Was this section right here. Now it's CAA due to the shift in the reading frame. And then it would be UAC. So how's that going to affect everything? Well, AUG, that's still the same, MET. What about GGU? What is that over here? GGU, that's going to be glycine. That's going to be the same, right? However, what happens after this? Let's look at what happens after this. CAA, GLN. So this is going to be GLN. And what about UAC here? UAC. This is going to be TYR, right? So that's the result of the insertion of a base, right? And you can go ahead and on your own, if you want to, go ahead and delete one of these bases and see what happens. Delete a base and see what happens here. I'm going to go ahead and erase some of this stuff here. Let's go ahead and erase some of these lines here. We'll get back to, because I want to show you guys a point mutation and how that can be silent at times. So let's say we have, I'm going to underline all of our triplets here. Let's say instead of inserting or deleting something, I end up swapping this G out for an A. This G gets swapped for an A accidentally. Well, what's that going to do to end here? You're going to end up with GGU. What's GGU going to be over here? G, if you follow it down, U is going to be glycine. Oh, we still got glycine here for the second one. And our reading frame has not shifted, which means that the following proteins should be the same. So in some cases, you can have what's called a silent mutation, right? Or a mutation which doesn't have a major effect on protein structure because it makes a similar protein to that it did before. Okay, so I do want to talk about a few other ways in which DNA can be changed inside of an organism. And for some of these, we're going to be talking exclusively about bacteria. For these next three are typically associated with bacteria. But transformation, if you remember that bacteria kind of has these little pieces of DNA called plasmids. This is going to be important to understand these little plasmids, right? Are especially in the next video here. If we have a bacteria, it's got its kind of chromosomal DNA over here. It's got this circular DNA, but it's also got these little smaller circular pieces of DNA called plasmids, right? It's also got these smaller circular plasmids. And that's what we're talking about typically when we talk about transformation, is the uptake of a plasmid by a bacteria, right? So this is taking DNA from the environment, and you end up with this DNA on the inside of these bacterial cells. This is called bacterial transformation. This is transforming a bacteria by inserting a plasmid. This is going to be very useful, especially in our next video when we talk about some of the ways we can create tools with this. Transduction is the introduction of DNA via some sort of viral vector, typically. So a virus can come in, inject its own DNA or RNA, and the bacteria can either reverse transcribe it or just incorporate that into its DNA and start making those proteins, right? This is transduction. Of course, this can happen in our cells also, right? Viruses can attack our cells as well. Conjugation. This is bacterial sex, right? This is when bacteria form kind of a link to each other, a temporary link. And one bacterium will kind of replicate its DNA, make a new copy, and ship it off to another recipient. This is the way bacteria kind of exchange genetic material and increase their genetic diversity. Now, transposition. This is something that does happen in eukaryotic cells. It was discovered by Barbara McClintock. Barbara McClintock. She discovered this in maize plants. But she discovered, and it was kind of interesting. It's still interesting today, but kind of revolutionary at the time, that genes have the ability to jump throughout the genome, right? These were called at the time jumping genes. Still called jumping genes, I guess. But genes can jump to different parts of the genome. They can be cut and pasted, or copied and pasted. They can be transcribed into RNA and then reverse transcribed back into DNA. And then a transposase, which is obviously ASE, an enzyme, can insert that DNA back into the somewhere in that organism's genome, right? Somewhere at a different location. So this happens quite often. It happens in our genes. It's something that's well known. It can make a genome size increase as well, so a genome can get bigger via this. And we do notice when we look at DNA, there's a lot of duplicate genes. This is a result probably of this transposition process. That's all I'm going to talk about when it comes to mutations and the way DNA can change in organisms. Join me in the next video for biotechnology.